Hi, I see I've got somebody logged on already. I'm just getting started. If whoever's already logged on can just say hi, let me know if the feed is okay, if the video and the audio is okay, if you can hear me well. Hi to the two enthusiastic and early birds who've logged on. Not sure who you are. I know I've got some friends tuning in for today to support. So if you'll just let me know in the comments if you're hearing me and seeing me okay, if the audio is okay, and who you are, so that I can say hi officially and welcome you. I'm also just going to be okay, so it's officially 1 o'clock. I'm not sure how many people are going to be logging in for today. I hope we'll have a few, as this is our first live stream, our first live video teaching session. Is that you, Nicole, that's already logged on? Great. Okay, so hey, yay, I've got Nicole logged on. Yay, thanks, Nicole. Yeah, I'm just gonna keep an eye on that number, Nicole, because I don't know, as I said, I have no clue who's gonna log on. And I'm gonna definitely give it a few minutes. Hope I can edit all of this out when I do the editing side of things. Based on the number of new subscribers to the channel, I thought we might get a few bodies rolling in, but I see so far it's just you. I'm just gonna kind of delay and hang on for a moment. I don't want to let go or launch until I see if we've got some actual students that are going to tune in. So yeah, I'm going to give it, I think maybe about 5 minutes, Nicole, because it's still just you logged in. Can you see from your side how many people are logged in? Okay, so you can also, okay, we've got some more people that have logged on. Hi, welcome to whoever's just logged on. Please let me know who it is, if it's some of my friends coming by for support or if it's biology students that are logging in. So just let me know in the comments who's just logged on. I'm also going to just be giving a few minutes to see how many extra people are going to log on. So I'm not going to get started straight away. I'm just going to wait. Hi, Kanye. I see you there. Welcome, welcome. Nice to have you. Thanks for coming around. Hope it's going to be more than just us few, but that's fine. Even if it's just for one, I don't mind. That's going to be awesome. Can you just let me know, darling? You're definitely in grade 12, and which curriculum are you following and which school are you from? So I just have a bit of a basic idea. And as I've been saying for the last few minutes, I'm not sure how many other people are going to join. I am just going to delay for a little bit and do a little bit of a chit chat whilst we wait and see how many others might be joining us. So I know there's 4 of us online at the moment. So the other 2, if you just let me know who you are, so I just have an idea of who's online so far, because I just want to see if we've got any other biology students logged on, because I know, Kanye, you've requested that we start with genetics today. So, hey, you might have scored the lottery. If there's no other biology students and nobody else has got any preferences, then you get it. It's going to be genetics for you. But so far, okay, just going to give it a few more minutes and then we're actually just going to start, because I don't want to hang around and wait for too long. Okay, wonderful, wonderful. That's great. Thanks, Kanye. That's perfect. I'm going to start telling you so long because, as I said, there's not, well, there's another person that's just joined us. Welcome. So for any biology students that are on, I'm going to just be starting off. I'm planning to do these sessions regularly. There will be another session this afternoon at 5 p.m. I'm hoping to schedule one for tomorrow, and I'm going to be doing this every week, even during the week. I'm not sure yet on the times. I'll advertise those as soon as I'm able to set up my schedule. But I'm going to be doing sessions throughout the week, all the way up to the exams. This is going to be ongoing. So for those of you that are on and you're attending schools, you're not homeschooled, please will you share the word, the info, the link, encourage your friends, your classmates to also come and attend these sessions. Because I'm trying to grow the channel and get YouTube to support me that way. Hi, Emihle. Nice to see you. Welcome. Grade 12 doing CAPS. That's awesome. I'm so excited to see you guys. Bear with me. This is my very first live stream session. I haven't done one of these before. I've just been creating some videos on YouTube. If you haven't had a chance to, you can always have a look after this. There are a couple of little things to go and check out there, but not much yet. I'm trying to get the channel going. So please spread the word to your friends, your colleagues, your classmates, whomever. Let them know that I'm going to be doing these live streams quite regularly until you guys write exams. I do have a student who is on the IEB curriculum. He is writing his exam, his first biology exam, on the 12th of November. So I'm going to be cramming in as much as I can to support you guys up until at least the 12th of November. It's going to be every weekend. I'm going to do sessions, and then I'm hoping to do sessions at odd intervals throughout the week as well. So let's get started, because I don't know if we're actually going to get any other bodies along the way. They can just fall in as we go. So Emihle, Kanye has previously asked me to start with genetics today. Is there any, can you both let me know which sections you're finding the most challenging and which you kind of think we'd like to start off with? Just give me a quick idea in the comments. Whilst you're typing that, I'm going to let you know that I am going to be working through the Nelson Mandela University text. It's a really awesome, I don't know if you're aware of it, if you've come across it, you can get it online, you can download it free. It's just such a beautiful summary. It's a very simple, easy to read and understand flow of the basic course material, and I find it's really nice to just help us get our head around the basics. Also, what I want to do for you guys at some point sooner rather than later is we're going to start going over past papers, but I do want to first focus on core material and then understanding the core material, and we'll throw things in as we go along, especially if you can log on regularly, because I'd like to have a bit of a format to what I'm going to be presenting. Okay, so let's get started. Any comments yet, Kanye and Emihle, or are we gonna jump in? Because I think what I'd like to do is I'm actually going to start at the beginning of the curriculum, which is going to be DNA, because I'm planning to do the entire curriculum. Hang in there, and we'll get to the parts that you need help with. Okay. Also, because there's not many of us, and this is a very small live stream at the moment, feel free as we go along, if you've got questions, just pop up a little emoji to say I've got a question, and I'll stop. Don't worry if you need to interrupt. I will give opportunities as I go along. Okay, ready. So let me just share the screen with you guys. and we can get started. Okay. Okay, so this is the curriculum that we're going to be following. As I've said, the Nelson Mandela for the Department of Education. And just a quick, we're going to look at the table of contents. I mean, you guys know this is what you've had to do for the year. DNA, I'm going to follow it systematically as we go through. So today we'll start with DNA, the code of life. Okay. Just excuse all the scrolling. Okay, life at molecular, cellular, and tissue level. So before I even carry on and actually launch into the content, I just want to also clarify a few things if you guys are going to be tuning in regularly. I'm seeing you there, Kanye. The part where you have to calculate offspring with the Punnett squares. I know that's going to be a bit tough. I'm not going to do that today, hun. I don't want to jump ahead because the way that the thing is set out is actually quite nice. So I want to try and keep it in a systematic order. Come and regularly log on and we're going to get to it as soon as possible, I promise. Also, what I like to encourage my students is please make sure you've got all your books, your notes, any resources or materials that you want to reference, have that ready and handy with you, have scrap paper, pencils, etc. lying around, because there may be things I ask you to do as we go along, and you know, you don't want to have to suddenly dash off and run around looking for stuff. So when you come onto the live streams, if you're going to make it at 5 o'clock, just have everything ready and rearing to go. Okay. So what I like to encourage my students as well, again, I'm not sure if you guys have started doing this or how you do this. Again, feel free to make comments so I get to know you a bit better. But I like to encourage my students to always have diagrams and pictures at hand, to work from pictures and diagrams before you even start reading, because that really is the essence of where biology starts, okay? It's so much easier to follow biology when you see a picture in your mind. And as I'm talking, if you can have a picture or diagrams ready to reference, to keep an eye on whilst you're listening to me, that just helps with getting an understanding going, and it helps you just get into the flow of things. If you're trying to just learn a bunch of sentences and words, it gets a bit, you know, it's exhausting because there's a lot of info in biology, okay? So always, when you're doing your biology, when you're working on your own, reference images, reference diagrams. And the very first starting point, it's a bit of revision, it's the cell. Okay, so let's just get there. Bear with me, guys, because I'm not used to this page sharing thing either. So thanks for coming along and being my guinea pigs whilst I figure this out. Okay, so we're going to start with a very basic revision of the cellular structure, because in biology, understanding the basics helps when we build on it and get more and more complicated in our understanding, in our concepts. Okay, so it's important to know the location and the function of the organelles within a basic human cell, because this is going to lead on to all of our DNA and all of our genetic stuff. It all just builds upon itself. Starting off with figure 1 on the screen, you can get a better diagram with a few more details, but this is a very nice basic starting point. We know that in plants, we've got a cell wall, but we're not doing the plants. So in the human cells, no cell wall. Okay, we just start off with a cell membrane, the outer layer of a cell. We know that the content within that cell, cytoplasm, which is pretty much the good stuff that everything floats in, okay, it's a base substance. All the organelles of the cell are suspended in the cytoplasm. It's a watery substance, and it allows for metabolic processes. I mean, that's the essence of a cell, metabolism, metabolic processes. The nucleus, wow, we all know that that is the powerhouse, right? So we need a nucleus. We need to know what the nucleus is doing and what is so important about the nucleus. Let me go and just move this down a tiny bit. Let me know quickly, guys, if the screen is okay. Can you read and see it adequately, or do I need to zoom in? Is it okay for you guys? Before I carry on, I just want you to give me an indication if you're able to view what's on the screen, okay, or if I need to zoom in. Okay. Whilst you're typing, I'm just going to a bit. Okay, so one of the most important components in the cell, we'll get to the nucleus in a second, but we've got things called ribosomes, okay? Tiny little dots that you can see there in the label. Ribosomes are very small round organelles, and they're normally attached to the endoplasmic reticulum. All of this is going to become relevant when we do protein synthesis. And they can be, the ribosomes can be free floating in the cytoplasm, okay? They are also sometimes found inside other organelles like the chloroplasts, which doesn't apply to human biology, and the mitochondria, but in very small numbers. Okay, they are the site of protein synthesis. So hence we need to know about them and where to find them. It's a big deal. And they consist of RNA and protein. Good. Okay. We get to what I call the brains of the operation. Okay, this is the powerhouse, the nucleus. It controls all of the cell's activities. So essential that we understand the role of the nucleus, because it's going to become important later when we do meiosis, right? A quick basic diagram there again. Remember, it's all about diagrams. Understand the labels and where everything goes, and that helps a lot. We start off with our nuclear membrane. Remember, our nucleus has 4 parts. The nuclear membrane is quite obviously separating the nucleus from the cytoplasm. The nucleoplasm is the stuff inside the nucleus, equivalent to the cytoplasm for the rest of the cell. And then right in the center, we've got the nucleolus. Of course, we've got the little pores called nuclear pores around the nuclear membrane. Okay. Remember that the nuclear membrane is doubled. It encloses the nucleus, and it's got tiny little pores that are going to allow things to come in and out of the nucleus. Again, it's got a jelly-like substance in there, the nucleoplasm, and the nucleolus is a dark body, which is suspended in the nucleoplasm. It's hiding in there. It's like all nice and cozy and comfortable until it's needed later on. It contains, very important to understand, start seeing the pictures in your mind, guys, because if you can see it and picture it, it just helps you when it comes to exam time. Okay, so free nucleotide bases. We'll get to what those are in a minute, but just make a mental note. And also another tip to give you guys, when you're doing biology, there's a lot of words involved, okay? There's a lot of words, English words to explain everything. But what you want to always look out for when you're doing biology are the biology terms or the terminologies. You want to go and make notes of those and pay attention to those and fit those words together, okay, to sort of filter out all the other English, as I call it. So just see these key words in your mind and start to see how they connect. Right, the nucleolus, it's suspended in the nucleoplasm within the nucleus, and it's going to contain free nucleotide bases. It produces ribosomes, so they get made in there. It's the factory. And then we've got the chromatin network, which is found in the nucleoplasm. This contains our DNA, which forms the chromosomes. And this is basically the recipe factory, right? The recipe of life. Because I consider DNA and genetics to be like baking a cake. It's all about a recipe. All the ingredients like that go into making a cake. Same as how do we make a human? All the ingredients. This is the process. So stick with me. I know sometimes it sounds a bit wacky, but I don't do things conventionally. So hang around and you'll see. So the DNA has the chromosomes in there. They contain the genetic code, obviously. We're going to figure out how that all becomes relevant later on. All good so far. Everybody still happy, awake, alive, etc. How are we doing? Let's see. Hi, Boosie. You good? Nice to have you. I think you've just maybe come on a bit, or no, now you've popped up. Great. The screen's okay. That's good. Just having a quick check on the comments. Everybody's still good. Are we still alive? Are we still awake? Are we good? Are we rearing to get this done today? Okay. Hope so. I'm going to carry on. I'm going to plow ahead. Okay, so let's talk about the structure of nucleic acids. Thanks, Nicole. All good as well. This side, phew, except I'm a bit hot. Bear with me. I'm under the lights here. So to start off with the key terminology that we want to look at, nucleic acid, monomers, and nucleotides. Let's understand what they are. So what is a nucleic acid? And if you guys would like to answer by way of helping you do your, testing your own level of where you're at, feel free to add to the comments. Don't be shy. If I'm asking a question and you want to add a comment or add an answer, that's great practice for you guys, okay? So nucleic acid is a type of an organic compound. Remember, organic life, inorganic is dead matter, non-life, okay? A monomer is the term for a basic building block, like bricks of a house or a wall. So when you're building a wall, you start off with your little bricks. That's your basic unit, the smallest basic unit. Same when we're talking about humans and all these complicated processes and structures, we got to start right at the beginning in the basic department, the monomers, building blocks. A nucleotide is a little building block or a monomer, which is going to form DNA and RNA. So in this segment or in this module, we're going to be focusing on DNA and RNA. Those are the two biggies. And to make sure that we understand the differences, because there's big differences, but only very subtle differences. Okay. We know that there's two types of nucleic acids, what we've just mentioned, organic compounds, the DNA and the RNA. I'm not pronouncing those long words, you know what they are. And together, DNA and RNA are going to form the basis of all life on Earth. I mean, literally no life exists without DNA and RNA. So we've got to understand this before we can build on our biological understanding and on knowing, you know, the complicated processes that are in biology. Okay, so let's get on and see what's nucleotides and what's all these other fun things. The basic structure of a nucleotide. So remember I said at the beginning, we have to always look at our diagrams and labels and pictures, because this helps us understand stuff way better than just a bunch of writing. Always start off with a picture. So there's a picture of a nucleotide. It's a schematic representation of what it's going on down there at the very teensy-weensy, you know, you can't even see the stuff. We know this. So picture this whenever you talk about DNA and RNA and nucleotides. Just see this in your mind to help you, you know, not lose your grip on the processes that we're going to be discussing. Because I know a lot of people get a bit lost with what goes where, what comes, especially the sequence of things when we start doing replication and protein synthesis. Okay. So the basic structure of a nucleotide is always a phosphate group, which is represented with the P, the luminous pink P. Our sugar molecule. Notice the phosphate group is connected to the sugar molecule. And then we've got our NB for nitrogenous base. You guys should already know at this point in your curriculum your nitrogenous bases for both RNA and DNA. Okay, quick question time. Hope you guys are still listening. Who can tell me which one of the nitrogenous bases is the one that changes between DNA and RNA? Because we know that we've got 4 nitrogenous bases in DNA and 4 in RNA. Do you guys know what they are? Can you remember? Just a quick word there from the guys that are doing biology. Or are you not sure of the names of the nitrogenous bases? Give me an idea of where you're at with this section. Okay. I'm going to carry on. If you guys are typing, you need to type faster. Okay. So a nucleotide, our basic little building block, is going to have these 3 components. Okay. And we're going to start off by doing DNA. And let's look at the specifics of DNA because, okay, uracil in the RNA. Well done, Emilia. Yes, it's going to be uracil in the RNA. And the equivalent in the DNA is thymine. Okay, so that's the one that's going to change. The others are all the same. So let's carry on and look at this. Our basic terminology, just to get our DNA brain going. Okay, so let's talk about DNA first. DNA, we know it's deoxyribonucleic acid made up of the nucleotides. There it is. Our nitrogenous bases are adenine, thymine, guanine, and cytosine. Okay, they carry the genetic code or the recipe for protein synthesis. So when we want to bake a cake, we go to DNA to get the recipe to tell us the ingredients and how to do it. We get a thing called nuclear DNA. So I want you guys to pay attention to this. We're going to see how it's relevant later on. Nuclear DNA, we know that's DNA found in the nucleus, but we also get extranuclear DNA. DNA found outside the nucleus. It's going to be in plants, obviously in chloroplasts, so we don't worry about that. Mitochondrial DNA. Okay, it's actually a nice exam question. So just make a tiny little note. There's 2 places where we're going to find DNA. We get a double helix for DNA. It's the spiral twist, okay, where we've got a double strand or 2 strands. And they are joined together and they're twisted spirally in their normal state. We're going to see, we know that they unwind and they untwist when they're going to multiply and replicate, okay? The term hereditary means genetic information passed on from a parent to an offspring. That's all it is. There is a video on my channel, guys, where I've gone and literally written and I've also spoken all of these terminologies for the entire syllabus of the Nelson Mandela University textbook. Okay, it's about a 36-minute video, and it literally is just terminology definition. If you want to go and grab that and have a look at it, because it's a nice summary and it just helps you get through the stuff, because the secret to biology is repetition. You can't study biology a day before the exam, you'll never get through. Okay, good. I'm not going to cover the basic history or the history of the discovery of DNA. Go read it. I mean, it's easily available. It's right here on Nelson Mandela as well. It is one of those classic sneakies that they like to put in the exam. That's always an extra mark waiting to happen if you get to know that stuff. It often comes up in an exam. Right, so we're talking about now the location of DNA. And what did we say? 2 locations in a cell, mostly in the nucleus. So go back to your picture of your plant cell, your human cell. Go back to the picture, see it in your mind. Our little outer cell membrane, the organelles floating, the nucleus in the middle. See that whilst you're listening to me talk, okay? Nucleus, boom, that's where most of the DNA lives, nuclear DNA. And then we get a small amount called extranuclear DNA, right? The mitochondrial DNA is very useful for tracing ancestry. So just keep that in your back of your mind, because we're going to look at that later on as well when we do genetics. Okay, now we get to the fun part. Everybody still alive out there? You've got to occasionally let me know that you're still with me and conscious. Thanks, Emilia, for adding the note there. Uracil and thymine. Remember, guys, focus on your biological terms and be very careful of your spelling. Thymine is with an E at the end, because we want to make sure we're getting our bio terms really well done and well spelled, because we don't want to lose marks in an exam if we get words wrong. Technically, we're not going to get marks. Okay, so well done. Thanks, Emilia, for that. Okay, so we said that the structure of DNA, double helix, which means there's 2 parts. Okay, on the left-hand side, we see it's a double helix. That's the normal structure of it. And when it's getting ready to do its thing and replicate and so forth, it's going to unwind, and we get that schematic representation. It's a simplified diagram. You guys have seen this. Notice the little pink circles are on the outside. So those are our sugars, okay? Our phosphates, and then our sugars are the purple houses. And we get A to T and G to C. That's our adenine, thymine, guanine, and cytosine. Okay, they're going to pair up like that. It doesn't matter which order they go in. On one side, they will always have their complementary buddy on the other side. So it doesn't matter if it's an AT or a TA, because they will always find each other, and so it will go. And this is basically how the recipe for life is made. Note that we've got AG, T. I mean, it's in any random given order. And of course, this is what differs from person to person and gives us all our uniqueness. Okay, let me scroll down. Notice here at the bottom, there's a weak hydrogen bond. Can you see on this diagram? We've got our guanine and our cytosine connected, our thymine and our adenosine. And between them, we've got a weak double strand or double hydrogen bond. That's going to become important later on. So DNA has a double helix. It's a monomer. The nucleotides link. These are nucleotides, remember. One nucleotide, pink, purple, and in this case, green at the bottom. And on the opposite side, pink, purple, and red. So that's one nucleotide connected to another. And they are connected through the weak hydrogen bonds that we've said. Okay. The phosphate and sugar molecules, notice this is an interesting thing. They're attached by strong bonds. Very subtle there that some people miss. So there's a weak hydrogen bond between the bases. That's the weakest point, if you like. But the phosphate groups and the sugars, okay, they are strong bonds, so that they're going to stay stuck during the replication process. We only want where the weak bonds are for the separation to happen. We'll see that coming shortly. Okay, there they are. We've said it now: adenine, thymine, cytosine, and guanine. The 4 nitrogenous bases for DNA. Right. We know which ones go with which, and it's always the same. You never ever get suddenly the other one joins with the other one. Biology is very organized until it goes wrong, I say. Because when it goes wrong, we get mutations and genetic defects and things, which we'll study later as well. Okay. The pairing of bases, which means A to whichever, A to T and G to C. The pairing of bases forms a long ladder-like structure, a long ladder. And we know that once they uncoil and they lie flat, that looks like a ladder, and it can be extremely long. Okay, let me just have a little, sorry for the noise, sometimes the phone goes off in the background. Okay, so DNA strands wind around proteins known as histones. Make sure you know what that word is. Okay, and I'm going through this quite quickly, and I'm doing a brief summary. If you guys need to, we can always do another session and go into things more detail. It's just that we've got a lot of info to cover, so we're just kind of getting an overview today. Okay, so we know that DNA carries our hereditary information, the stuff that's going to get passed from parents to offspring. The hereditary stuff is going to come through in the form of genes, and genes are short sections of our DNA, just short sections of DNA, coding for a specific trait, which means my hair color is genetic, unless I go and get some dye at Dis-Chem, you know what I mean? Eye color, all of the physical things that we see are decided by our genes, okay? And we don't have control over that. So we also, interesting to note, our blood group is genetically coded, which is why, I don't know if you guys know that if you have a blood transfusion, they have to very carefully check what your blood is and make sure they match the correct blood to you. Otherwise, it has drastic consequences. It can even kill you. And there's also been they've discovered a gene linked to breast cancer. So ladies that are born with certain genetic recipes are prone to develop breast cancer, even if they lead a healthy lifestyle. So some ladies actually go and have themselves tested if there's a lot of history of breast cancer in their family, and then sometimes take preventative measures before the breast cancer can develop. Okay, so even behavior of an organism can be determined by DNA. Certain, especially in the animal kingdom, we see a lot of different things that are very, passed on, instinctive behaviors and things. Okay. DNA strands do not code for anything and are known as non-coding DNA. That's most of the DNA strands. Okay, so scientists are still checking out the importance of this non-coding DNA. It's an interesting concept. We'll touch on it more later. But let's look at the main functions now of DNA. It controls the functioning of cells. The actual cell themselves are controlled. It regulates and the functioning of our genes, and it passes on hereditary characteristics. Excellent exam question right there. When you guys are studying your biology, also try and make note of and kind of say to yourself, if I'm an examiner, what am I going to ask? What are the main functions of DNA? Ding, ding, ding. Easy question for like 3 marks. Okay, so try and always keep that in mind. I'm not going to go through the activities in this case. You guys can always go and look at that if you download the Nelson Mandela yourself and you've got your own activities to go through. Let's move on to RNA. Okay, so let's note the differences here, as already mentioned. Well done. We've now got uracil replacing thymine. Okay, that's the distinct difference. So for RNA, it's still the nucleotides, the nitrogenous bases are found. You see, we've still got the adenine, the guanine, and the cytosine. The only one changing is the uracil, so don't get that confused. And then with RNA, we've got 3 types of RNA. Messenger, it's the mRNA, which carries the code for protein synthesis. It's going to go into the nucleus, get it from the DNA, and carry it to the ribosome and say, yoo-hoo, I went and took a copy. It's like a photocopy machine, okay? It will go in there. It'll take a look at the original, the official version of things. It'll make a note, okay, ding, 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 make a copy, ding, off to the ribosomes. Yoo-hoo, here it is. We get ribosomal RNA, which now is where the protein synthesis is going to happen. It's going to be made there. And our transfer RNA, that's like the taxi system, okay? It's going to go and fetch all the amino acids according to the recipe, and it's going to bring it to the ribosome to make the protein. So it's like a whole little factory setup going on. Everybody has to work together with everybody else. Okay. So those are our 3 types of RNA. Interesting that they are formed in the nucleus by the DNA. So you can see the relationship and how everything is interconnected. Okay, so at this point, I need a swig of water. Any questions? Everybody else okay? Still alive? Are we going too slow? Are we going too fast? You guys got to give me some feedback here, because this is my first live stream. I'm used to teaching privately, and it's a bit different when you've got regular feedback and interaction with your students. So feel free to give me some feedback, guys. I won't be upset. whilst I'm waiting for some feedback, I'm just going to reiterate, guys, because I like to repeat things over and over again. And if that gets too boring, you also need to let me know. But remember, when you're doing biology, always have diagrams and figures and pictures in at hand, your textbook, your notes, or whatever, so that when you're reading all of this stuff, like I'm reading this stuff off of this Nelson Mandela, you've got to have pictures to refer to all the time, just to keep it, to help your brain see the pictures. Because if you see biology in pictures, it really makes it a lot easier to when it comes time for the exam. If you can sit in that exam room and they start asking you a question and the picture pops up in your head, you can almost see it and then help yourself go through, you'll see when we do exam techniques, it helps a lot. It makes it a lot easier. Okay. So if you guys are still with me, I'm going to carry on. The location of RNA. We've said the messenger RNA, it's formed in the nucleus. Okay. Still good. Thank you so much for that, Emmy here. Happy days. Thanks for that. At least somebody's with me and awake. Yay. Okay, cool. Messenger RNA. Messenger RNA. Now think about what we were saying. Picture the cell. Picture the little nucleus within the cell. You've got your endoplasmic reticulum and your ribosomes in the cytoplasm on the outside. Okay. Messenger RNA is formed or made in the nucleus. It's made in the cell powerhouse, the brains of the operation. It's made in there. Okay, and then it's going to travel through the nuclear membrane, through the nuclear pores into the cytoplasm, where it's going to go and attach to the ribosomes. Okay, ribosomal RNA, it's found in the ribosomes in the cytoplasm. So see that in your mind. Think to yourself, where is the cytoplasm? Yes, there it is. Where are the ribosomes? Yes, see it, because this helps you remember this stuff. Okay, and the transfer RNA, it's the taxi. Think about the taxis in South Africa. They just go wherever they want. They hoy there, all over the place, right? So the transfer RNA is found freely in the cytoplasm of the cell, missioning around, doing its thing, okay? Structure. Let's do structure of RNA. Okay. We know that, like DNA, RNA is made up of nucleotides, our monomers, our basic building blocks. And they come together to form longer chains called polymers. Poly meaning many. Polymers. Again, try and remember what goes where in order. Mono becomes poly sequence, okay? RNA is a single-stranded structure, and it's not coiled. The sugar, remember let me just move the page down for us quickly, so we can see there, okay? The sugar in RNA is ribose, and in DNA, it's the opposite. It's deoxyribose, okay? So remember, they've all got a phosphate on the outside. It's a strong bond connected to its sugar, which is represented with the little purple S. And then we've got our little nitrogenous base, which is going to connect through weak bonds in the DNA. I hope we're not getting confused. Okay, so the phosphate and the sugar are attached to one another alternately to form the chains. Can you see in this image, and why I say pictures are so important? If you've just read all of that, but you didn't look at the picture, it isn't necessarily that obvious what it's talking about. But notice there we've got the little P, the little pink P, okay? It's connected to the S, and that S is connected to the next P, which is then connected to, and so it goes, okay? So it's an alternate connection, and that's what's going to form the chains, the polymers. Let me move down a bit. Sorry about the sharing of the screen is quite tough because I've got to keep jumping around, so bear with me with that. Okay, we've said it already. Just to recap, uracil, not thymine, okay? That's one of the key perfect exam questions. They love to catch you with that. 3 types of RNA are important for protein synthesis. Now we're going to compare DNA to RNA because, again, perfect exam question. You guys have to make sure you get this solidly in your brain because they love to do this. This is why I love the Nelson Mandela, because if you look at how beautifully they put this, it's also nice and simple and easy to understand. It's just really well put together. So go grab it or come and watch this video if you can tolerate me. Okay. So we know that DNA and RNA are very similar, okay, almost the same in a lot of ways. Let's look at the similarities, because in your exam, please, guys, these are super, super important words to pay attention to. If they ask you for the similarities and you just don't even read the question properly, you go banging ahead and you give the differences, well, you just got a big fat zero because you didn't read the question. So always make sure, are they asking you for the similarities or are they asking you for the differences? Okay. They both, RNA and DNA, both have sugar, okay, alternating with a phosphate. So that's important. We know that they have 3 nitrogenous bases in common, adenine, guanine, and cytosine. So these are part of the similarities, and they both play a role in protein synthesis. Perfect. Most of the time, the exam question is on the differences, but don't fall into that trap and forget to read, because Murphy's Law, this exam, they're going to ask you for the similarities. Okay. Now, let's focus on the differences. The main differences. Nice little tablet format. So the sugar in DNA is the deoxyribose sugar, and in the RNA, it's the ribose sugar. So they do have sugar, but now we've got to differentiate. It's deoxyribose and ribose. In our DNA, we know we've got the double coiled helix, like the twisty chippy, like the Fritos chips. Go get a bag of Fritos chips when you study this, and munch on that when you're talking about DNA to help you remember. It's the twisty chippy, okay? And for the RNA, it's a single strand. It's not twisted. For our DNA, we know that we've got the thymine, and the opposite is the uracil. That's the differences for these guys. Only, they say here found only in the nucleus, but remember that's not true. We do have mitochondrial DNA, which is not in the nucleus, but it's a tiny amount. And we know that RNA is found in the nucleus, in the ribosomes, and the cytoplasm. So RNA is kind of all over the place. Good. Are we still there? Cool. Okay, we've been going for 40 minutes. What I'm going to do with you guys is I'm just going to see if I can push through and finish this section, and then at the end we'll have some quick questions and answers, and then we'll probably finish off the session if that's good for everybody. Okay, so hang in there. We're doing okay, I think. Right. So now we're going to talk about DNA replication, and replication means basically making more, as in making a copy. Okay, don't get confused, guys, when we talk about this. It's not going to be the same as when we get to meiosis later on, which is a different thing. So we're now talking about DNA replication, as in making more of itself. And it's the process where DNA makes an identical copy of itself. Okay, so it's like a photocopy machine. And it needs to be identical to retain health and, function and structure within a body. This is the stuff that goes wrong when we start seeing things like cancer happening, where we start getting mistakes and errors in all of these processes. And then we start getting things like cancers and diabetes and all of those weird things. Okay, so it occurs during interphase of the cell cycle in the nucleus, and we'll see more about that. But just see the picture in your mind, okay? It's in the nucleus during interphase. We know that pictures, you know me, by now you know me, pictures, right? It's all about pictures. So let's go. Let me get my pictures. So now we're going to be doing the DNA replication. Okay, so now it's time to make photocopies, and this is where it gets a bit more interesting. Well, I think so. DNA, double helix, your Fritos chippies. Take a munch and start the process, okay? In figure 7a, we see the first thing is that it unwinds, it unravels, but it's still stuck together, okay? It's still stuck together, but it's now lined up. And notice the purple strips representing the outer part of the nucleotides. We've got our bases represented in the middle there with our weak hydrogen bonds, okay? Step 2, the weak being the key word here, because remember the other bonds are the strong bonds. They're going to withstand this process. They're not going to get broken in this process. The weak hydrogen bonds are going to start to break like a zip. When you're pulling a zip up on your jacket or your jeans. Zip. So we're going to start to see the strands are going to separate. They're going to unzip. And it's a perfect analogy. It really is very easy to understand at this point. Notice, though, as soon as it unzips, you've technically got where you had an A, it's now on its own. Its buddies moved off to the other side. So where we had A and T stuck together, dunk, they're going to separate. Now we've got an A here and a T here. So we know that they're going to need to attract their opposites, which are going to be the free nucleotides floating around that are going to come and connect. And that's going to lead us to have a double replication. We'll see it's like a photocopy effect. We know how this goes. Okay. More scrolling down. Come on, screen. Okay. We now end up in figure 3, where we've got each original DNA strand. So remember, this is the original DNA strand on the left and the original on the right. Those are the ones that have separated from the original. Now, notice we've got a whole bunch of funny little pieces down below with a little T, a little G, a little A. Those are the loose individual nucleotides, the free nucleotides that are floating around. Okay, so we've got our template or our recipe to say this is what it needs to look like. Okay, and we now need to go find the partners or the friends to come and complement to build another portion. The free nucleotides are going to build the DNA strand, okay, and it's going to attach to the original. Once we have that happening, the complementary nitrogen bases have linked up as normal, and you can see in figure D, we've now in essence got 2 of what we started off with. It's identical now. I hope that's simple enough. I mean, it is fairly straightforward and self-explanatory. Just look at the pictures when you study this. It really helps make it a lot easier to understand. The result is we've got 2 identical DNA molecules, okay? Each consists of 1 original plus 1 new strand. So we've got 1 original and a new strand, 1 original and a new strand. And boom, we end up with 2 identical. But we've refreshed somewhat. You know, we've kind of freshened things up a bit. That's the basic concept here. Okay, it's important for cell division, and this process is called mitosis with a T, mitosis, because we haven't halved anything like we are going to do in meiosis. We are just replicating what's there. Because remember, our body cells die off and they need to replace. You know, there's different cycles for each of our body cells. You know, the eyeball cells have to refresh themselves. You can't have, these are not the same cells that you were born with. You know, your cells have to keep replicating to refresh themselves as we age and as we go through life. So the chromosomes are copied, and each new identical daughter cell produces the same number and type of chromosomes. That's DNA replication. Okay, let's talk about quickly what errors can occur. We've already touched on it a little bit, but we know that errors can occur because, you know, nothing in life is perfect. So during DNA replication, when this whole process is happening, the splitting, the new joining of the new bases, mutations occur, okay? A change in that nitrogenous base sequence. Because if your genetic code said we need a G, an A, a T, and an A, and somehow that got switched around during one of these processes, it's suddenly not the same as it was. It's not a correct, true copy, and you're going to have what we call mutations. Sometimes things can be added, sometimes they can be deleted, or the sequence is disrupted and changed around, and it results in the change in a gene structure. And eek, we know that that's not cool. Okay, again, some activities that you can see if you go through the Nelson. These are very nice to go through, so go and download this, guys. You can get it online for free. It's really easy. Nelson Mandela University. Okay. DNA profiling. Okay, let's move on to that. DNA profiling. Very interesting. And of course, a little contentious, I think, sometimes. But a DNA profile is simply a pattern that's produced on an X-ray film. It consists of these little lines that we see, okay? And you can see in the example here in figure 8, the lines are in different places. Some of them are a little thinner and some of them are a little thicker. And these are unique to each of us, except for identical twins. Okay, that's very interesting. And I'm sure you've seen some programs where there's you know, some controversies because, oh, maybe it was his twin or whatever. Okay, so the pattern is different lengths, different thicknesses, and in different positions. Okay, and this is very, very useful for a number of different reasons. We use these profiles, and this is a very good exam question. Make notes. Good exam question. What do we do, or why do we use DNA profiles? Easy to see. It's summarized for us to identify crime suspects. We know how DNA works. They take DNA. Either you did the crime or you didn't. Is it your blood on the scene or sometimes semen? They can tell because we're all unique. Okay. Also used to prove paternity or maternity, to prove who mom and dad are. If we've got an issue with a child and we're not sure who the mom and the dad are, they will do paternity tests and maternity tests to see. They can also determine if we do DNA profiling, it can determine the probability and the cause of genetic defects. As I was mentioning earlier, there is a gene that they've shown can lead to breast cancer. So before you get the breast cancer, if you go have the genetic test, they can say to you, listen, this was very likely that you will develop breast cancer later. And then also, when people are needing an organ transplant, we know that you can't just go and whip out somebody's kidney and give it to the next one, because it's very specific and it's got to be a match. So they establish if the tissue types of the two parties are going to be compatible. Can you have this person's heart or this liver or this kidney? And they do the DNA profiling to test that. Okay, and also to identify relatives. Like if they find somebody who's had an unfortunate death, and they're not sure who this person is, but they suspect it might be a certain person, then they'll ask the relatives for their DNA, and they'll do a comparison to see if this person is related or not, and they can often determine who the unidentified person is that way. Okay. It's normally very reliable. It's generally considered very reliable. However, the interpretation and the comparison of profiles has to be approached with caution because of the following. And again, I've seen this in past papers, a very nice exam question sitting right here. Okay. Humans interpret the results, which means mistakes can happen because we just are mistake prone, we're not perfect. So humans are going to interpret the results. They could unfortunately mismatch. The method of profiling differs from laboratories, which produces inconsistencies. Also, during the DNA profiling, they're only using a very small piece of DNA, because remember, we've got chains and chains of DNA that are incredible. These recipes are no joke. They are very complex. So we can sometimes only have a little piece of DNA that we found for testing, and it's not 100% unique to a particular individual. Because remember, my little portion could actually be the same as your little portion. It's only when you go lower down the chromosome that you notice the differences. And if it so happened that we were testing those small portions, oops, well, then they could point the finger at the wrong person because we only tested a tiny piece of the DNA. And so it's always got to be backed up with other stuff. Okay. And profiles can reveal information that could be used against them in a prejudicial way. Like when we do HIV testing, genetic abnormalities, some insurance companies don't want to get involved, and you can read that. It can be contentious. But those are the reasons why DNA profiling can be, a little non-reliable. Okay, so there's an activity. This is a classic in the exams where they ask you to say if somebody is the suspect or if somebody is the mother or the father. Make sure you know how to do these activities. I'm not going to cover that in detail now, okay? Okay, I think we're still ticking along. I just want to see how much more we've got left here. Protein synthesis. Okay, guys, I think I'm going to stop the session now because it's been going on for quite a while. I'm going to carry on with protein synthesis in the next session. If you guys miss it, the video will be online at some point, but I'm going to stop here because I'm also tired. So thanks for sticking with me and for hanging in there. Any questions? Let's see if you've got any questions. If there's anything that I've discussed you want to just clarify, or hold on for a moment. I'm just going to wait a little bit. If you guys are busy typing up any questions, I'm happy to answer. So I'm not going to end the stream immediately. If you don't have any questions, you're welcome to log off at this point. Make a note if you're keen. I'm going to be doing another session at 5 p.m. today, and then I'll schedule for tomorrow. I'll just need to check the time for tomorrow, but I will try and do a session tomorrow. Boosie, I didn't see you mentioning in the comments, love, where you're from, which school and which curriculum you're following. Are you also doing CAPS? If you can just let me know. Just going to wait a few more moments. If you guys don't have any questions, then I'm going to end this session. Okay, so there we are. Thanks for coming. Thanks for being here. Thanks for being my guinea pigs. I really appreciate it. And I'm hoping to see you guys again. And yeah, we'll get you there. Biology's easy. We just got to be consistent. Thanks again. Cheers, guys.